Good afternoon, good morning, wherever you are. And thank you to the organisers for the opportunity to speak about vaccine boosting of SARS-CoV-2 COVID-19 vaccines, a very hot topic at the moment. I'm Penny Moore. I'm an associate professor at the University of the Witwatersrand and the National Institute for Communicable Diseases in South Africa. So I think where we've begun in terms of administering vaccines is in a sense phase one or crisis mode. The idea here was to build immunity as quickly as possible, and we did that by very rapidly deploying vaccines that had been developed very quickly. But we are now entering a slightly different era, I think, in terms of SARS-CoV-2 vaccines, where we're beginning to worry about whether and when we need to boost. We've known for quite some time that after we build immunity, whichever way we choose to build immunity, that the durability is unknown and that the protective titer is unknown. And so while we initially focused very hard on building the immunity, the question now is when and how do we boost in order to keep people protected? But I wanted to take a little step back and remind everybody that although in many parts of the world we are thinking very hard about when to boost and thinking about third and fourth and even fifth boosts, in large chunks of the world, including the part of the world that I live in Africa, we're still in phase one. There's really a substantial disconnect, depending on where you are in the world, between how well different countries are vaccinated. And while, for example, North America has already achieved pretty good levels of vaccination, large regions in Africa are only predicted to have vaccines administered from 2023 onwards. So really a very big disconnect depending on where you are in the world. In terms of thinking about when one might need a boost or an additional dose, I think there are really three main scenarios, and I'm going to go through them all in the slide and then break them down for the rest of the talk. The first of these is the emergence of new variants against which the existing immune responses are no longer effective. And to some extent, we've already dealt with this, but I'll speak a little bit more about it. The second real area where people have been talking about boosters is in populations in whom the primary vaccine series failed to elicit a sufficiently robust immune response. And then the third group is the general population, everybody else really, who developed very good responses to the primary vaccine series, but in whom the responses have now started to wane. And the question is, at what point have they waned enough that we need to administer a booster? So let's think first about the emergence of new variants. As Delta emerged across the world, I think that was a major concern for many people. Delta is now dominant, as many of you will know, across the world. And the question really was whether the emergence of this variant, which showed relatively high neutralization resistance, would impact vaccine effectiveness. But actually, as the data has emerged, it seems that vaccine effectiveness has somewhat been reduced, but remains incredibly high, particularly in the context of severe disease. So ranging from protection from infection is somewhere between 40 and 84%. But against hospitalization is somewhere between 75 and 95%, depending on the vaccine. And that's shown here in this schematic, which compares the vaccine effectiveness against Alpha, which was relatively neutralization sensitive, to that of Delta, very neutralization resistant. And you can see that there's actually not a big difference either in the Pfizer or in the AstraZeneca vaccines. And that seems to be true for other vaccines too. It may not continue to be true. It is entirely possible that more resistant variants may emerge. And indeed, some already have, though at this stage, none have outcompeted Delta. I'm just showing you a couple of examples here. This is Mu, which is dominant in Colombia. You can see even if Mu is here in blue, and even compared to Beta, which was the most resistant variant that had been described thus far, Mu is on par, if not more neutralization resistant. And here's some data from our group showing resistance of a new variant called C.1.2, which has been detected in South Africa. C.1.2 shown in purple here compared to the original D614G. And you can see that this variant too is pretty neutralization resistant. So we will have to watch these variants, and they may continue to impact our vaccines. Let's think about populations in whom the primary vaccine series has failed to elicit a sufficiently robust immune response. This is largely people who are immunocompromised. And this is a very diverse group of people. It includes people who are on long-term steroid treatment, who are on biologics for autoimmune conditions, individuals with hematological or immune malignancies, solid organ or bone marrow transplant recipients, people on renal dialysis, or folks with primary immunodeficiency or disorder, and of course, HIV positive individuals. And I'll speak individually about these two groups. Many of these groups have already been shown to have compromised responses to the flu vaccines. And actually, focusing on this group of people is both important, very important for the individual who is at high risk for COVID-19, but also important for the community, because this is the group of folks in whom variants of concern may arise if these individuals are not able to clear the virus. So really an important group to focus on. There's lots of data, unfortunately, now in the field showing that people with various immunosuppressive conditions do respond, in many cases, quite dramatically less well to vaccines. This is data looking at individuals with chronic inflammatory conditions, either who are on immunosuppressive treatment. If you look at this graph here, which looks at immune responses after the second vaccine, and you compare people with chronic inflammatory conditions in the open circles with those with the healthy controls in the full circles, you can see that in general, although it's not dramatic here, those individuals with chronic inflammatory disease seem to do, in many cases, less well. On the flip side, on the plus side, they did in fact mount a response, many of these individuals, and it seemed to be safe. But clearly these vaccines are less effective in some of these individuals. And that's particularly true in individuals who are on certain treatments, for example, methotrexate shown here, which seems to dramatically reduce people's ability to mount vaccine responses. This graph compares healthy individuals, 98% of whom mounted responses, with individuals who do have inflammatory immune disorders but are not on methotrexate, in which group 91% of people mounted responses. But look at the massive difference in people who are on methotrexate, only 50% of whom actually mount a response. And if you look at the medians here, you can see it's significantly reduced. And I've just copied some text here from this paper, which I think sums it up very well. It's that patients on methotrexate, and this applies to other regimes, may need alternative vaccination strategies, such as additional doses of vaccines, dose modification of methotrexate, or even a temporary discontinuation of this drug. And this applies, as I said, to other drugs such as cancer treatments. There's also significant evidence in people who've received transplants, solid organ transplants. This is one such study that shows reduced humoral responses to the Pfizer vaccine in individuals who received kidney transplants. And you can see here on the left controls, and on the right-hand side, with much, much lower responses, individuals who had received transplants. All of the controls developed a positive response to the spike protein, but only 38% had a positive response, or 38% of transplant recipients had a positive response. And not only that, but again, the mean titers were much, much lower in transplant recipients. The question is whether a third dose can boost responses in these individuals. And there's mixed data about this, and I think it's very early. Mixed data in terms of regimens, primes, boosts, very small numbers in many cases. It does seem that in some cases, people who failed to mount a response to the first and even the second vaccine can mount a response to the third vaccine. But I think this data is a little concerning in a small group. But 2 weeks after a third dose, although the low responders to the second dose were boosted, of the non-responders, people who had failed to respond to the second vaccine, 2/3 still had no antibodies, despite a third boost. And it will be worth our while, I think, thinking about whether vaccines are in fact the right way to protect this particular group of people, or whether we need to consider monoclonal antibodies in these cases. Let's change gears a little bit and think about people living with HIV. This is close to my heart. I'm, as I said, based in South Africa, where 7.5 million South Africans are HIV positive, and more than 2 million of them are not suppressed, not virally suppressed. In other words, not on ARVs for whatever reason, or the ARVs are not working. In my country, the adult HIV prevalence in people aged 15 to 49 is over 19%. So in my country and in many other countries, whether HIV positive people respond to vaccines is really a huge issue, and actually very little is known. This is some data looking at a small randomized double-blind placebo-controlled phase 1b/2a trial that was conducted in South Africa of the AstraZeneca vaccine, a trial that was led by Shabir Mahdi. And in this case, it did seem that binding to SARS-CoV-2 spike was similar at day 0 to day 28 and day 42 in either HIV negative people or those individuals living with HIV. But very little known for other vaccines, and even less for people who are viremic, who are not on ARVs. In general, these trials tend to recruit people who are very well controlled. So I think we have much to do to understand whether people who are living with HIV have reduced responses. In studies that we are doing looking at HIV positive people who are infected, not vaccinated, we definitely see reduced responses and a delay in those responses. And I think it'll be important to understand whether that translates to vaccine responsiveness as well. The third group, of course, is everybody else. This is the general population who developed a very good response, but in whom the response to the primary vaccine series has waned over time. And this is something that's been hugely discussed in the press in the recent weeks and days. We're only now learning really about durability of the SARS-CoV-2 COVID-19 vaccines. And the reason for that is we've only had them for a few months, and one can only understand durability by studying people who have received the vaccine. The early efficacy data for the RNA vaccines certainly suggested durability past 4 months. This is the Kaplan-Meier curves from both the Moderna vaccine on the left-hand side and the Pfizer vaccine on the right-hand side. In both cases, the line at the bottom is individuals who received the vaccine. And you can see that at 4 months, this is 128 days post-vaccination, you can see that that line is still pretty much flat and people are not becoming infected. So that looked promising at 4 months. And although the efficacy is perhaps lower in the adenovirus vectored vaccines, the initial data for these also suggested durability out past four months. However, we are now starting to look at what happens later because people have been vaccinated for longer. Certainly in this study, there's a significant reduction in humoral immune responses 6 months after Pfizer vaccine administration. This was a study that was done in nursing home residents on the right-hand side and controls, in many cases their carers, who had received a vaccine, either people who had been infected prior to receiving the vaccine or those who were infection naive. But regardless of which group you look at, you can see that if you compare the red to the blue, there's a dramatic drop-off in neutralization responses in this group. And I think we're seeing this in more and more of these kinds of studies. There's also emerging evidence of waning from the Israelis. Israel's been an incredible case study in a sense because the Israelis managed to get high levels of vaccines out very, very quickly early in the pandemic. And many people have been vaccinated, revaccinated with the second dose, and in fact now increasingly vaccinated with the third dose. This is data looking at people who received two doses of the Pfizer vaccine, so the standard Pfizer vaccine regimen, but partitioning them by when they received that vaccine. In other words, how long it's been since they received the vaccine. And if you look on the left-hand side, look at this, the group of 60 plus individuals, you can see that the rate of infection, so the higher the bar here, the more the number of documented SARS-CoV-2 infections, you can see that individuals who were vaccinated in January, shown in black, have a much higher chance of becoming infected than somebody who was vaccinated in May, so more recently. The same is true here if you look on the right-hand side for severe disease, much lower odds because the Pfizer vaccine protects extremely well against severe disease. But again, you can see quite a clear pattern, I think, indicating that the longer it is since you've had your vaccine, the higher the chances are that you either become infected or become ill. So I think this is this is one emerging data, and there seems to be more and more of this kind of data coming out that suggests that vaccine effectiveness may wane against Delta, because these are Delta studies, within 6 months. So similar data emerging from the United Kingdom that also suggests that protection afforded by 2 doses of Pfizer and AstraZeneca vaccines similarly wanes within 6 months. So of course, this does make us think about when and how often boosting will be required. Is there evidence that a third boost enhances the responses? Absolutely. There's good evidence for this already. This is a study that came out very recently in the New England Journal of Medicine that looks at what happens after a third dose. These investigators had samples from dose 1, dose 2, and just before dose 3, and then looked to see what happened after the administration of a third dose. And you can see this is looking at serum neutralization titers. You can see that 7 days after dose 3, there's a massive increase in neutralization titers, and that these titers remain high 1 month after dose 3. Whether they will remain high for 6 months or a year is something that we don't yet know. And so that is one of the key questions, is how long the neutralizing antibodies and other immune functions will be retained after a third boost. And there's similar data that has been described from the Johnson & Johnson trial, in this case after the second dose of Johnson & Johnson, because remember that's a 1-dose regimen, that also shows good boosted responses in that vaccine. There's also data from Israel that suggests that a third boost also enhances protection, not just neutralizing antibodies, which seems so far to be a very good proxy of how well a vaccine is doing, but not absolute. This is data that has also recently been published in the NEJM that shows that 20 days after a third boost, there is a 20-fold reduction in confirmed infection rates, suggesting that there's almost immediately excellent protection from infection after a third dose. Very much what we would expect given what we know from the second dose of this vaccine. What would the boost be? This is something that is very much undefined at this stage and the subject of many studies. In many cases, people seem to be suggesting the administration of the same vaccine that you received in the primary vaccine series. So if you received 2 doses of Pfizer, you would get a third dose of Pfizer. If you received 1 dose of Johnson & Johnson, you would get a second dose of the Johnson & Johnson vaccine. But of course, mix and match or heterologous prime boosts are becoming increasingly popular. This is Angela Merkel, who received Moderna as a second jab after the AstraZeneca shot. She was an early advocate of mix and match. But there's increasingly many studies starting to look at this. This was one of the first, looking at a heterologous prime boost for the AstraZeneca followed by the Pfizer vaccine. You can see here the graph on the left-hand side shows you a comparison of people who received Pfizer as a prime and boost versus those who received the AstraZeneca as a prime and the Pfizer as a boost. And if you compare these 2 sets of neutralization data up here, I beg your pardon, RBD binding data, you can see there's no significant difference between the 2. And I think we have to recognize that in real life, this is going to become increasingly common, particularly in areas where vaccines are scarce and hard to access. People will simply take whatever is available. Is there any reason to think that prior infection impacts vaccine responses? Absolutely. Lots of data now on this, and I'm going to whip through this because I think it's pretty well known. This was an early study from Leo Stamatatis and Andy Maguire looking at what the effect of prior infection was on vaccine responses. Here you can see that if you compare this group here of people who were previously infected and then received a second dose or even a first dose of an RNA vaccine, you can see very, very high neutralization titers, much higher than what you would see in the case of people who received 2 vaccines but had not previously been infected. So prior infection, which essentially acts as another vaccine in this case, certainly boosts antibody responses dramatically. in our study here in South Africa, we've seen the same with the Johnson & Johnson vaccine. In people who had no prior infection, we do see low-level neutralizing responses after a single dose. But in people who were previously infected and then received the Johnson & Johnson vaccine, we see massively increased neutralization titers. So as for other vaccines, and this has been well established now for the RNA vaccines, adenovirus vectored vaccine also drives much higher neutralizing antibodies after prior infection. And this is something that may be very important in countries like mine, where up to half of our population has already been infected. So in conclusion, there's a really strong ethical need to equitably distribute vaccines. It's massively disconnected at the moment. And while some countries are routinely already implementing third doses and thinking about fourth doses, many, many millions of people have yet to receive a first dose. And I think this is something we need to be constantly aware of. The variants do seem to reduce the effectiveness of vaccines against infection, but not against hospitalization. And increasingly, we need to work out which should be our focus. Hospitalization clearly is the first priority. But as we move forward in this pandemic, we will need to think about how to protect against infection, not just hospitalization, in order to protect our most vulnerable. The data is very clear that immunocompromised people are in urgent need of additional doses or other modalities, such as monoclonal antibodies, in the case of people who simply do not respond to vaccines. Prior infection definitely significantly boosts immunity, and that's something that we can harness potentially in countries where we're struggling to get vaccines out. But there are many questions that remain about additional boosts. How long will they last? How many will we need? Is this something, if durability is going to come crashing down every 6 months, will we need boosters twice a year? Or will a third dose push stronger responses that will last for longer, as it's been seen in other vaccines? Are there safety risks? At the moment, we have very little information about the administration of the third dose. Though I think it's clear from all of the data that key populations, as well as the general population, will need these. We have a lot to learn still about boosters. And there I will stop and thank the people in my lab for many conversations on this subject. Thank you.